Since failure is not an option, we need to figure out what to do next. And we will try to do this by learning a little bit of biology. And biology will give us a hint on how we should implement our algorithms. Some of you may find description of somewhat convoluted biological concepts that I am about to present difficult to digest. If you feel like this, don't worry. If you believe me that DNA replication is an asymmetric process, then you can simply skip this part and go to the point when we derive from this biological knowledge how to design efficient algorithms. So the first thing to remind ourselves is that DNA strands have directions. And the two strands of DNA run in opposite direction. Here on this slide, the blue strand runs clockwise, and the green strand runs counterclockwise. And if you were a DNA polymerase, how would you replicate a genome? If I was a DNA polymerase, I would do something very simple. I would wait until DNA unwind a little bit, recruit 4 DNA polymerases, and we'll just move them along the genome trying to replicate it. It looks like just 4 DNA polymerases are enough to replicate the whole genome. And when replication fork enlarge, I continue the replication process. This is simple, but completely wrong. And if there are biology professors attending this lecture, they are probably already writing a petition to fire me and send me to Biology 101 camp. The reason why it doesn't work is that DNA polymerases are unidirectional. They can only copy DNA in the direction that is opposite to the direction of DNA. Which means that when we want to recruit 4 polymerases to replicate DNA, 2 of them, this one and this one, will be working just fine. But 2 others won't be able to move because they cannot move in the same direction as the direction of DNA. Then we can classify DNA strands into 4 half strands. The strands that I showed you, the blue strands that goes from origin to terminus, is the reverse half strand. And I have no problem replicating it because moving from origin to terminus goes in the opposite direction to DNA. Likewise, this thick green line that I showed right now also doesn't present any problem replicated. 1 DNA polymerase can accomplish it. But 2 other half strands present big problem because they cannot move in the same direction that they go. So if you were a unidirectional DNA polymerase, how would you replicate a genome? Here's a potential solution. Wait until fork enlarges. And when it enlarges, start replicating it in the opposite, in the same direction as DNA. Then fork enlarges a little bit more, you put another DNA polymerase and continue replicating. 4 DNA polymerases won't be enough because each of these DNA polymerases that I recruited copied just approximately 3,000 nucleotides. So you may need a huge number of DNA polymerases to proceed this way. You can see that the resulting fragments, these many fragments that are being built called Okazaki fragments, complicate our life a little bit. But in the end, after this process is over, we will have many Okazaki fragments, so we will have genome copied in many little fragments. And the only thing we need afterwards is just to repair the gaps between different Okazaki fragments and to replicate the genome. So the only thing we need to learn about this process to proceed with our algorithm afterwards is that reverse and forward half strands, so thick half strands and thin half strands, have very different lifetime. The reverse half strands lives a double-stranded life because it's constantly replicated. There is hardly a moment when it lives double-stranded. But the forward half strands spend a large portion of its life single-stranded, because it has to wait until replication forks open until it starts replication. This is, I hope this is clear, but there is one looming question. Why would a computer scientist care? And we will learn in the next segment why computer scientists should care about this. So asymmetry of replication affect nucleotide frequencies. Why? Let's think about this. Single-stranded DNA has a much higher mutation rate than double-stranded DNA. That's why if one nucleotide has a greater mutation rate, then we should observe a shortage of this nucleotide on the forward half strand because it spans single-stranded life. Which nucleotide, A, T, C, or G, has the higher mutation rate, and why? It turned out that there is a peculiar statistics of mutation rates of nucleotides, and cytosine, C, rapidly mutates into T. What is quite amazing is that through this diamidation process, rate of mutation rise 100-fold when DNA is single-stranded, which means that the strands that live single-stranded life very quickly get depleted from C. What does it mean for us as algorithm designers? Forward half strands that live single-stranded life has shortage of C and normal G. Reverse half strands that live double-stranded life have shortage of G and normal C. Now, keeping this in mind, let's take a walk along the genome. We start at terminus, and let's say we move from terminus, according to the red line, to origin. In this case, we move along the strand at which there is C is high and G is low, which means that G minus C is decreasing as we walk, right? But when we walk along this half strand, on this half strand, C is low and G is high, which means G minus C, total number of nucleotide G minus total number of nucleotide C, is increasing as we walk. Again, this sounds like peculiar and not very important thing. Why do we care? I ask one more question. If you walk along a genome and you see and you count number of G and number of C that you minus number of C that you saw, and you have seen that number of G minus number of C have been decreasing and then suddenly start increasing. Just imagine you walk through the genome, you count the difference G minus C, and it has been decreasing and then suddenly start increasing. My question is, where in the genome are you? And to figure out where in the genome are you, we need once again to look at peculiar statistics. The only place in the genome where behavior of G minus C switches from decreasing to increasing is origin of replication. Which means that if you walk along the genome and see that G minus C has been decreasing and then suddenly start increasing, it means you just passed the origin of replication. And this is the hint for our algorithm. So let's define mathematical framework for how to deal with this. We define skew of K as simply the number of G minus number of C in the first K nucleotides of the genome. And skew diagram is simply a plot showing skew of K against K. Now let's construct skew diagram for the E. coli genome. It looks beautifully. Where do you think is the origin of replication in E. coli? Something that we almost had no hope to find. Of course, it's at the point where it was decreasing and then suddenly start increasing, which is here. We found the replication origin in E. coli. And now, let's try to find frequent word in this origin, and hopefully we will find DNA box in this way? Well, after we run our frequent words problem, unfortunately, it turns out there are no frequent words that appear even 3 times in this replication origin, which means that we failed. There are many reasons why we may fail. Maybe origin of replication as derived from Skew diagram doesn't show precisely to the area where we want to see. Or maybe, just maybe, we don't have a good grip on how hidden messages in E. coli origin look like. Should we give up? Let's try to proceed further and figure out what else can be done. And when we were looking for frequent words, our view of frequent words was very naive. We assumed that frequent words are simply k-mer. But maybe hidden messages that cell uses to initiate replications are more elusive, more subtle. Let's look at this origin of replication in Vibrio cholerae and try to see. Maybe there is something else. We already found 6 9-mer in this region, but maybe, just maybe, there is something else that deserves our attention. And if you look carefully there, then you will see that in addition to these 6 hidden messages, there are 2 more. There are actually 2 9-mers that look almost like canonical 9-mers. They differ just in a single mutation. And they also represent DNA boxes because DNA can bind not only to the perfect 9-mer, but to different variations of this 9-mer. So to find this type of more elusive frequent words, we need to solve frequent words with mismatches problem, when the input is a string text and integers k and d. We want to find all most frequent k-mers with at most d mismatches in the text. And finally, after we've done this and run our frequent word with mismatches problem, we do find somewhat more elusive frequent words in E. coli genome. And they turn out to be, indeed, real DNA boxes in E. coli. Now, what I described is a very idealistic view of how replication can be found. In reality, some bacteria have fewer DNA boxes, and our frequent words algorithm won't work for finding them. Terminus of replication is often not located directly opposite to the origin of replication. And the Skew diagram is often more complex than in the case of E. coli. And that's why we want to show you some open problems that relate to origin of replication, and I will describe them in the next segment. So many open problems left for finding origin of replications, even in bacteria. We wanted to design research projects that would be able to introduce you to open algorithmic challenges in this area. And that's why we feel that our course is not only MOOC, massive open online course, but also MOOR, massive open online research projects. And we were very lucky. We have recruited some of the top bioinformaticians to direct your research projects. The first project will test the old dogma of bacterial replication. As I told you, bacteria have a unique single replication origin. But recently, in 2012, ARCHAEA conjectured that maybe it is wrong. And in particular, if you look at this Skew diagram for the genome that lives in the guts of the tsetse fly, you see that maybe, just maybe, it has 2 replication origins. But there is no definite proof yet. And we want you to develop computational tests that may help us to figure out whether this or any other bacterial genomes actually use 2 replication origins. It would come quite handy for bacteria because they would be able to replicate themselves faster than bacteria that just use a single replication origin. The second project is also directed by Professor Gelfand. In this project, we want to find origins of replication in ARCHAEA. You know that life consists of 3 domains: bacteria, eukaryotes that include us, plants, yeast, and the third strange domain of life that is called archaea, that do not look like bacteria, but do not look like eukaryotes. And it is known that in archaea there are multiply origins of replication. This, for example, in this Skew diagram, these origins of replication have been confirmed. However, there is still no algorithm and no software tools that would allow one to predict how many origins of replications are in archaea and where they are located. So this will be our second research project. Our third research project will be directed by a leading expert on origin of replication, UNIDIRECTIONAL at the University of Sydney. If you feel that bacterial replication is complex, wait until you hear about replication in yeast, let alone human. Yeast have hundreds of origins of replication. They have to work somehow coordinated with each other to avoid disasters during replication. However, there are still no algorithm or software tool for predicting origins of replication in yeast, and UNIDIRECTIONAL will direct the research projects aimed at solving this problem. And our last problem relates to the questions that we asked in the beginning of the course, but never answered. Remember, we asked, is it statistically surprising to find a nightmare appearing 3 or more times within a random text consisting of 500 nucleotides? This seemingly simple problem turns out to be surprisingly difficult. It turns out that different k-mers have different probabilities of occurring in the string, in a random string in particular. And for example, 0-1 string have very different statistical properties than 1-1 string when it comes to its appearance in random Boolean text. This phenomenon is known as overlapping words paradox, because different occurrences of 1-1 can overlap, but different occurrences of 0-1 cannot. So in this problem, we want you to answer this question that is shown on the top of this slide and figure out, give exact probability, how surprising it is to see frequent words of different types in DNA strings. And this project will be directed by Professor of Mathematics at University of California, San Diego, Glenn Tesler. Our first lesson is coming to an end. And you notice that in this lecture, we hardly talk about developing new bioinformatics algorithms. Instead, we were focusing on formulating bioinformatics problem. And it's a feature rather than a bug. In bioinformatics, developing new problem formulations and transforming sometimes convoluted biological reality into elegant algorithmic problems is at least as important as solving bioinformatics problem. Indeed, the key difference between an algorithms class and a bioinformatics class is that in algorithmic class, the problem formulations are often written in stone, and students either learn that the problem is intractable or learn how to develop algorithms for solving the problem. In contrast, the bioinformatics class starts from problem formulation, and students learn how to translate very difficult biological reality into elegant algorithmic problem. In this course, we will learn that the problems that first look like intractable can be turned into relatively simple problem by simply reformulating them. Happy Rosalind, and see you at the next lesson.